Hello everyone. Today we are going to discuss DNA repair mechanisms. Now, in order to understand the importance of DNA repair mechanisms, we must first appreciate that DNA holds the blueprint of any living organism. It is therefore not just critical but vital for any organism to pass on the information in an unaltered manner from one generation to the next. At the same time, it is also important to understand that there might be changes or mutagenesis that may be induced during the process of replication. Changes that are good for the cell, that is, the changes which are advantageous, may accumulate for hundreds of years to cause what is called as evolution. However, those changes which are detrimental for the cell may lead to the death of the cell or some kind of impairment which are extremely dangerous for the cell. One such example is that of various types of cancers. DNA repair mechanisms help in the process of preservation of this information. The process of changes that are brought about by a process called as mutagenesis, which might be the topic of our discussion in some other lecture. The mechanisms of DNA damage repair are broadly classified into two basic types. The first type are those types which occur during the process of replication or before the process of replication. So we say that they are pre-replicative, or those mechanisms that occur before the process of replication is actually complete. The second set of mechanisms are those mechanisms which are employed once the process of replication is over. So many a times you can refer to the post-replicative changes also as the recombinational repair systems, or those repair systems which occur in a recombinational mechanism. So to begin with, we will start with the first type of DNA repair mechanism, which is pre-replicative in nature. Now the pre-replicative DNA repair mechanisms are again classified into two types. The first type is direct reversal. A direct reversal mechanism basically means that if there is a damage in the DNA which is led from A to B, then a reversal will mean that B is simply returned back to A. So basically there is no excision of DNA, there is no removal of any part of the DNA, and what we see is the change that has occurred is simply reversed back to normal. Now this is one of the very important mechanisms, and the process by which this mechanism is brought about is seen here in case of the formation of a thymine dimer. Now if you see here, there are two nucleotides that are present here. The two nucleotides, both of them contain a T or a thymine nitrogenous base. So both of them are thymine containing nucleotides that are joined together by a phosphodiester linkage. Now in such a sequence of DNA which has two thymine sequences present adjacent to each other, on exposure of UV light there may be a reaction which may cause formation of bond between carbons of the adjacent thymine groups. Now you see that this is one thymine, this is the second thymine, and between the two thymines there are carbon to carbon linkages which are formed, which leads to the formation of a cyclobutane ring. A butane refers to the fact that there are 4 carbons in the ring structure. Now this cyclobutane ring, which is formed between two thymines, what it has essentially done is that it has impaired the two thymines from forming a natural hydrogen bond which it should have formed with another strand in front of it. This might be clear with this photograph, where you can see that the two thymines which are present, they have been joined together, and therefore they are not able to form a normal hydrogen linkage with the adenines which are in front of them in the complementary strand of DNA. Now in order to repair this kind of damage which is caused by UV light, a simple UV light triggered enzyme called as photolyase has been identified and isolated from bacterial cells. Now this enzyme, which is also triggered by the UV light, causes photoreactivation. When I say it causes photoreactivation, what it is doing is, in presence of light, it is bringing back the change to normal. So all it is doing is that in presence of light, it is breaking the bond between the two thymines and making sure that the thymines can form original and normal bonds with the adenines of the complementary DNA strand. At this juncture, it is also important for us to appreciate the fact that photolyase, the enzyme which catalyzes the process of breaking the thymine dimer and returning the thymine dimer back to the original form of the DNA, is activated by the blue range of the spectrum. Now, this information is of particular importance. If you need to study it further, you might find a reference where this enzyme may be of evolutionary importance and it might connect to certain eukaryotic enzymes, especially of the plant kingdom, which also interact with light and which are triggered by light and bring about certain developmental changes in case of the plants. Coming back to our portion of topic, so this is about the first type or the first category of DNA repair mechanisms, which are direct reversal, which come under the pre-replicative mechanism. The second type of pre-replicative mechanism is of excision repair. Now, excision repair can further be classified into three types. The three types of excision repair mechanisms include base excision repair, nucleotide excision repair, and mismatch repair. Let us take all three of them one by one, beginning first with the nucleotide excision repair. Now, nucleotide excision repair is basically removal of the nucleotide which is damaged by means of an excision. Now, what we have to remember in all these processes that we are going to discuss now, that is nucleotide excision repair, base excision repair, as well as mismatch repair, is that wherever there is a damage, that part of the DNA will be removed out, that is, it will be cut out from the original DNA, and that part of the DNA is going to be resynthesized. So, beginning first with how a nucleotide is excised and removed. We again take the example of a thymine dimer. A thymine dimer is typically formed, as we just discussed, a thymine dimer is typically formed as a result of UV light exposure. Now, a UV light exposure which has caused the formation of a thymine dimer, I want this part of the DNA to be removed, and once this part of the DNA is removed, I want the same part to be resynthesized. Now, a set of three proteins are required to identify this problem and remove the lesion or the damage that has occurred in the DNA. Now, if you look at this, there is a thymine dimer which is formed in the first set of DNA as represented here. On either side of this lesion, there will be two proteins that are attached which are called as UVRB and UVRC. Now, UVRB and UVRC, they will be associated with the 3' and the 5' ends respectively, which means that at the 3' end there will be UVRB and at the 5' end there will be UVRC. You can remember this very simply because 3 comes before 5 and B comes alphabetically before C. So 3 is associated with B and 5' end is associated with UVRC. So once this UVRB and UVRC are associated with the 3' and the 5' sides of the damaged part of DNA, they excise the DNA in coordination with the third protein, which is the UVRA. Now, UVRA, UVRB and UVRC, sometimes they are together called as the exenuclease. Exenuclease stands or represents the fact that these three proteins are working as excision proteins or excision enzymes. So basically, they are excising a small part of DNA that has underwent a change. So UVR A, B and C together, they identify this lesion containing DNA strand, they split it or they cut it from either sides and they bring it out with the help of an enzyme called as helicase. Now, to understand what the helicase exactly is doing is that you might have seen a chocolate or a lozenge, right? So what do you do to bring out the lozenge? You just untwist it. So what helicase is doing is that there is a loose part of DNA. In order to bring it out, you just twist it. When you twist it, the loose part of the DNA, it is dislodged from the main part of the DNA. So coming back to this, once the helicase acts, the loose part of the DNA is removed and this part or this gap which is thus created is then healed with the employment of a DNA polymerase and a ligase. DNA polymerase will synthesize, it will create a new strand, and the DNA ligase will seal the gap which is formed as a result. The use of both these enzymes or the functions of both these enzymes have been discussed in the DNA replication section. The next type of excision repair is the base excision repair. Now, base excision repair is normally employed in those cases where there is some kind of a chemical alteration in case of the base, that is the nitrogenous base of the DNA. Now, the primary type of chemical modification that might occur is a deamination kind of a modification. This deamination modification may take place on exposure to certain chemical agents, which may lead to the conversion of a cytosine either to a uracil or an adenine to a hypoxanthine. Now, normally a cytosine getting converted into a uracil or an adenine getting converted into a hypoxanthine, both of these changes are chemical changes that need to be repaired. However, out of the two of them, it is the first type, that is cytosine getting deaminated into uracil, which is more dangerous and more critical and requires to be repaired more readily. Why? The reason is cytosine normally has a complementary base pairing with a guanine, that is C with three bonds, three hydrogen bonds. It pairs with a guanine on the complementary DNA chain. When a cytosine gets converted into a uracil, the complementarity behavior of uracil is different from cytosine. So a uracil would normally want to pair with an adenine. The second point is, usually uracil is not present in a DNA, so a uracil containing DNA is subject to some kind of repair. Now, this repair is more urgently required because it has changed the nature of complementarity of the DNA strand and therefore changed the basic theory on which the process of replication will be based. In order to repair this kind of a damage, we first need to identify the DNA portion which has underwent a change. We can identify that very clearly because of the mismatch that has occurred and the presence of a uracil in a DNA. So a U containing DNA, which is formed by the deamination of a cytosine containing DNA, is subject to this kind of a repair. The first enzyme that is required for this repair to take place is a DNA glycosylase. A DNA glycosylase is an enzyme that will break the glycosidic bond present in the nucleotide. Now, where is this glycosidic bond? Let me remind you all that in the structure of DNA, you might have studied that a nitrogenous base is linked to the sugar molecule by means of an N-glycosidic linkage. So basically what the DNA glycosylase is going to do is that it is going to remove the nitrogenous base, that is the U in this case, from the rest of the chain. So the sugar and the phosphate, which is the part of the backbone of the DNA, will remain intact. However, the uracil, which is only the base attached to the sugar, will be removed by means of DNA glycolase. Now what happens when you remove this base only? When you remove this base, you create an AP site. AP site, AP stands for an apurinic or an apyrimidic site based on what base has been removed from this position. So glycosylase has just removed a modified base, a base that has been modified by some chemical damage. The removal of this base has led to the formation of an AP site. Now this AP site will be recognized by the second enzyme of the repair mechanism, which is the AP endonuclease. An AP endonuclease identifies and creates a nick on the backbone of DNA. When I say it creates a nick, it means that an endonuclease breaks the phosphodiester bond between the two adjacent nucleotides. So it breaks the bond between the adjacent nucleotides at the AP site, removes this apurinic or apyrimidic sugar molecule from this site, and therefore you get a small gap in the portion of DNA which was damaged because of a chemical modification. Now this gap will be healed or it will be filled by the DNA polymerase, and the resulting gap will be sealed by ligase as usual, a process that we have seen time and again in replication as well as in the previous type of repair mechanism that we just talked about. So what is important that you all notice is that when we talked about nucleotide excision repair, we talked about a single step mechanism where this entire section of DNA was removed at a time, and then the DNA polymerase filled the gap and ligase sealed the gap. However, in case of base excision repair, we see that the same process of removal of the DNA strand almost takes place in a two-step mechanism, where first the base which has underwent the chemical change is removed. That site at which the base was removed is then identified, cleaved, removed or excised, and then the new DNA strand is added. So first you take out the base, then you take out the rest of the sugar, and then you fill the gap with the help of DNA polymerase and the ligase enzymes. Now, coming back to the third type of excision repair, which is the mismatch repair. So as the name suggests, the mismatch repair system detects and excises mismatched bases in the newly replicated DNA. Now at this point, there are two things that we need to remember and understand very clearly. This mismatch type of base and the repair of this mismatch type of base normally occurs as a proofreading activity of the DNA polymerase itself. Now DNA polymerase, as we all know, it is that enzyme which carries out the process of replication. Now while the replication is occurring, simultaneously along with replication, there is a proofreading activity during which such mismatches, if they are incorporated, they will be repaired. However, there might be certain odd matches or mismatches which might be left behind in spite of the proofreading activity of the DNA polymerase. It is those mismatches which will be repaired by this system of repair. Now this system of repair works almost simultaneously with the DNA replication, which means that while the DNA replication might be occurring in one part of the DNA, the other part of the DNA may be subjected to a repair system which might be a mismatch repair. Now, what happens during this mismatch repair system is something that we need to understand very clearly. A mismatch is a wrongly added base. So there might be an A in front of a G or a C, or there may be a T in front of a G or a C which is not normally present. So a G linked to an A is not a normal complementary base pairing, or a C linked to a T is not a normal or a simple complementary base pairing. It's an unnatural base pairing, so it needs to be removed or recovered. Now the process of recovery is not that simple. Now, when I have a T in front of a C, let us say, the first thing that I need to understand is, out of the C and T, which one is wrongly added and which one is the parental sequence. Now, is C a sequence which was present in the parent, and is T the base which has been wrongly added, or was T present in the parental DNA and is the C is the one which is wrongly added to the DNA during replication? To understand this, the first thing that this repair system does is it distinguishes between an old strand and a new strand. Now, as I just told you, this process of repair is occurring almost simultaneously with DNA. So the strands that are formed, the old strand and the new strand, they can still be differentiated from each other based on one chemical property. And that chemical property is the property of methylation. Now it is important for the students to remember that methylation is a process that naturally occurs in adenine base pairs on a DNA. Now, this process of methylation usually occurs after the process of replication is complete. Now, since the replication is already taking place simultaneously in one part of the DNA, the rest of the DNA which is just synthesized is yet to be methylated completely. So if I look at the strand of the DNA which is about to get a mismatch repair, one of the strands will be methylated, which will be the DNA strand, which will be the parental DNA strand, and the other strand which is yet to be methylated is the new strand which has just been synthesized by the polymerase. So it is this base which is present on the new strand which needs to be removed. So this repair system, which is working on the basis of three proteins named as MUT-S, MUT-L and MUT-H, the first thing that they do is they identify the position of mismatch. Now once they identify the position of mismatch, they adhere to this position of mismatch and they identify a particular site or a particular sequence on the new strand of DNA, and that particular sequence is GATC. So wherever there is a GATC nearest to the mismatched base pair, at that point MUT-H, MUT-S and MUT-L, specifically MUT-H, causes a nick. A nick is a cut. Now DNA is cut at the GATC site. So basically it has identified the nearest GATC site to the lesion, and MUT-H has acted like an endonuclease. Now this cut which has occurred at the nearest GATC site, it will lead to the excision of that part of the DNA which has been cut up to the mismatch. Now this process of cutting the DNA and excising it is catalyzed by the MUT-L and MUT-S together. The resulting gap will be filled up by DNA polymerase and sealed by ligase as per the tradition. Now, these are the pre-replicative, or let us say those processes of DNA repair mechanisms which take place simultaneously along with the DNA replication, or they might be occurring before the process of DNA replication. The next set of repair mechanisms are those that occur post-replicative. So these post-replicative DNA repair mechanisms, they usually take place in case of exposure to certain chemicals which might take place after the process of replication has completed. Now such conditions which might trigger damages in DNA, they may either trigger a single-stranded break or a double-stranded break in the DNA. The first post-replicative repair system that we are ought to look at is the single-stranded break repair. Now a single-stranded break repair system is something that is not commonly observed in case of bacteria. In fact, it has been reported and studied exhaustively in case of the human cells. Now for a single-stranded break repair, the primary protein, the primary enzyme that we need is called a PARP. A PARP stands for poly-ADP ribosyl polymerase. So it's a poly means multiple, ADP which is adenine containing dinucleotide, ribosyl, of course, ribosyl dinucleotide, and it will be a polymerase. So it is basically a polymerase enzyme which will only be associated with adding the A containing nucleotides. Now, to understand what is happening in this process, let us just look at the process very simply. Now suppose that you have a DNA. The first thing that this enzyme is going to do is that it is going to bend the DNA and expose that region of the DNA which is having the damage, which means that if this is the part or this is the region which has underwent a single-stranded break, it will be exposed further by bending the DNA, therefore creating or accentuating the gap which has been created by a single-stranded break. Now, once the DNA is bent and the gap has been exposed, PARP employs certain accessory proteins. With the help of these accessory proteins, what happens is, one of the ends of the DNA is extended. Now the extension will occur as per the 3', 5' rule of extension that normally occurs in case of DNA replication also. So suppose that this is the single-stranded break, this is the 3' end, so this end will be adhered by the PARP and other accessory enzymes and it will be extended. So when it is extended, it will kind of extend under the other strand of the DNA, and it will almost dislodge it from the normal complementarity, and the the resulting gap which is created after a particular sequence of bases, the resulting gap which is created will finally be sealed with the help of a DNA ligase. So what is happening here is that the 3' strand or the 3' end of the break is being extended under the 5' end. Approximately 12 to 13 bases length of this 5' end is taken off or dislodged, and it is removed or cut out, and the resulting gap is sealed with the help of DNA ligase. Now, study of this PARP and the accessory proteins that take part or that participate in this kind of a single-stranded break repair is extensively done with the relation of cancer and cancer biology, that is oncology in case of human cells. However, in other cells, single-stranded breaks may also be repaired in a recombinational mechanism, which usually takes place also in case of the double-stranded type of repair. So a double-stranded break repair is also sometimes referred to as the recombinational repair, because this is the mechanism also employed in case of the recombination of chromosomes. So a double-stranded break is healed or recovered with the help of the complementary strand which is still intact. Now let us look at the process in detail in a stepwise manner. The entire process can be understood in 3 different parts. Let us take up the first part first. Now suppose that the DNA has underwent a damage, and the damage has caused a double-stranded break in the DNA. Now a double-stranded break means that the DNA has got no reference for the correction. The reference can only be sought in the same sequence or a homologous sequence of the sister region of another DNA. So a sister DNA, with the help of the sister DNA, the double-stranded break will be repaired. In order to repair this double-stranded break, the first step is to cut the edges, which is technically known as end resection. End resection is nothing but biting off the ends of the DNA so that there is a 3' overhang. When I say overhang, it means a small region of single-stranded DNA which is not paired. So what has happened is a double-stranded break had created 2 portions of DNA, both had equal edges. Now, one of the sides or one of the strands of the DNA will be cut off. Which side or which strand will be cut off? In a break from 5' to 3', I mean that strand which is available to be cut from 5' to 3' end will be cut out, so that the single-stranded end that remains behind is oriented towards its 3' end towards the break side. Now, we have this 3' end towards the break side because it is this part which will be extended further, or which will be developed further with the help of a polymerase. Now, after the end resection has taken place, something very remarkable takes place. This 3' overhang of the damaged DNA almost invades into the homologous region of the sister DNA by taking out one of the strands and falling and start replicating on the complementary strand of the sister region. Now, if you look at this last step carefully, you will see that the black strand or the black region which is getting continued over the red region or the red strand of the sister DNA, this is the DNA which has suffered the damage. I mean, the black DNA is the one that has suffered damage. It is copying that part which it is missing from the homologous region of the sister DNA. Once it has copied this region, it will again be dislodged from this sister DNA and it will go back to its original position, and the complementary base pairing of the undamaged DNA will be restored as usual. Now, once this sequence has been copied and it goes back to the original, the black DNA goes back to the original position, the next part of the repair may be of 2 types. The two types of repair that might take place from here on are synthesis dependent strand annealing, that is SDSA, and double stranded break repair, that is DSBR. Let us first look at the B part of the screen, and let us first concentrate on this first type of repair system or sub repair system. Now, once one of the strands of the damaged DNA has been copied from the sister DNA, the next thing that may happen is it can go back to the normal position, and once it goes back to the normal position, it can itself act as a template and cause the synthesis of the other strand of the DNA, so no further crossing over will be required. So one of the strands has been synthesized, and based on this synthesized strand, a new strand will be synthesized and annealed in the usual position, with no further crossing over. This is the first situation. The second situation is when both the strands are synthesized by a crossing over. Now recollect what we have just seen in the previous stage. We saw that this DNA, the black DNA, which has dislodged one of the strands of the undamaged DNA and got itself copied. Now what is interesting is that the just dislodged strand of the DNA, which is complementary to the other strand of the damaged DNA, can also be copied at the same time. So simultaneously, both the strands of the DNA can be copied by dislodging the undamaged DNA in one go. So what happens is the 3' overhang, it dislodges the undamaged, one of the strands of the undamaged DNA. One strand of the undamaged DNA acts as a template for this part or the first part of the damaged DNA strand, and the second part it acts as a template for the second damaged strand of the DNA. So both the damaged strand take the help of or want a template in the undamaged strands of the sister DNA. Now this location or this junction that is formed here is an H junction, and the resolution of this junction is known as an HJ resolution. So this method is of a crossing over, or this method is of a recombination that might also help in the process of recovery of the DNA which has just suffered a double-stranded break. So these are the two types of, or two different categories of the DNA damage repair system that we have just seen. We have seen the pre-replicative changes and we have seen the post-replicative changes. Now, there is a third type of replicative change that we are yet to discuss. Now, the third type of DNA repair mechanism which has been understood very recently is a translation synthesis. A translation synthesis actually occurs side by side with the process of DNA replication. Now, while it is occurring side by side with the process of DNA replication, we might call it pre-replicative. However, this translation synthesis does not include any kind of direct reversal, or in fact it does not involve any kind of excision repair. In fact, this translation synthesis is not a process which is involved in repairing the DNA as such. It is almost a mechanism by the help of which you can bypass a damage. Now, again, translation synthesis can be of two types. Let us first understand what happens in a translation synthesis. Now, suppose that the DNA replication is occurring, and while the DNA replication is occurring, the replication fork meets a portion of DNA that has a lesion, that has some kind of a damage in it. Now, when it has some kind of a damage in it, there are two choices for the replication fork. Ideally, the process of replication may be aborted at this point wherever it finds a lesion or wherever it finds a damage in the DNA which it cannot bypass. However, aborting the process of replication will be of severe consequences to the entire cell. Therefore, this lesion is either avoided or it is replicated by a bypass mechanism. The bypass mechanism which is used to replicate this kind of a lesion, it can again be of two different types. The first type is a translational bypass, wherein wherever there is a lesion, that part of the DNA is not copied and the replication fork simply jumps over it. Now, when I say it simply jumps over it, it means that the DNA replication will continue in almost a template independent manner. At the point of lesion on the mother strand or the template strand, the DNA will not be copied in a template dependent manner, and the replication fork or the DNA polymerase which is carrying out the process of DNA replication will jump over this lesion and it will carry forward the rest of the process of DNA replication as usual. The second method by which you can avoid a lesion to take place is the strand which meets with the lesion. The daughter strand which is being synthesized or which has just met a gap, it cannot find a template. It reaches for a template in the other daughter strand which is being synthesized on the sister template of the same mother DNA. Now, you can see that this green strand did not meet with any kind of lesion. The green strand is the daughter strand which is being synthesized on the other, or it is being synthesized on the complementary strand of this lesion containing mother DNA strand. So therefore, this blue strand which is met with a lesion, it can find a template on the sister DNA strand, be copied there, and then come back to the original DNA or the original template of the mother strand and carry on with its further process of replication. So there are two methods of having this process. You can either have a lesion bypass where you simply avoid copying the damaged part of the template, or you copy the damaged part of the template from your sister which is being copied on the other strand of the same DNA. So that is translesion avoidance or translesion bypass in that kind of a synthesis. So basically what we have seen today is the various types of DNA damage repair, and to sum it all, we have seen that the DNA repair mechanisms can broadly be classified into those systems which occur during the process of replication or pre-replicative, and those processes of DNA repair which occur post-replicative. The pre-replicative type of DNA repair can be classified as direct reversal or excision repair mechanism. Excision repair mechanism can further be classified as base excision repair, nucleotide excision repair, and mismatch repair. The post-replicative repair, on the other hand, can be classified as single-stranded break repair and double-stranded break repair, as we just saw in case of the cells. So with this, we sum up the portion of DNA repair mechanisms. The references that have been used to describe this video or portray different parts or different reactions in this video have been enlisted in the description section of this video. What is also enlisted in the description section are those references which might help you to study this topic further and enhance your knowledge with regards to DNA damage repair system, as well as what changes or what modifications in the DNA damage repair system are associated with different diseases, especially cancer. So with that, we finished with the description of DNA damage repair, and this was a brief description with respect to this topic. I hope you appreciate this. There is also a feedback form at in the description section of this video. You can log on to that Google form and leave back your feedbacks on this video. Thank you, and I hope you keep studying this topic further.